When you think about verifying the food you eat, what's the first thing that comes to mind? For a lot of us, it's probably a stamp of approval, maybe a certificate hanging on a wall. But what if I told you the real story of food integrity isn't written on paper at all, but in DNA? To really get how that works, we need to look at a system a lot of us just take for granted.
So let's take halal verification as an example. From the outside, it looks pretty administrative, right? A stack of paperwork, a bunch of checks, and finally, a certificate. But step inside the laboratory, and that simple paper trail, it transforms into something way more powerful: undeniable molecular evidence.
And here's why that shift is just so critical. A system that only relies on paperwork has a huge, fundamental flaw. It's blind. It literally can't see the microscopic stuff like cross-contamination or fraudulent mixing that happens at a molecular level. All it can see is the paper it's printed on. But science gives us a whole new way to see. It actually borrows a concept straight from forensics. We all get that every person has a unique fingerprint, right? Well, it turns out every single species of meat has something just like that, a distinct, undeniable DNA signature. This gives us a way to do incredibly precise molecular identification.
So how in the world do we read that signature, especially if there's just a tiny, invisible trace of it? Well, that's where PCR technology comes in. The easiest way to think about it is like a molecular photocopier. It can take one single invisible strand of DNA and just amplify it millions and millions of times until, boom, it becomes a clear, detectable signal. This incredible power, the ability to find a needle in a molecular haystack, that is exactly what makes PCR the absolute gold standard for evidence-based verification.
All right, so let's put these two approaches right next to each other. The old standard, it's all about administrative paperwork. The new scientific standard, it's all about PCR and molecular evidence. This completely shifts our detection level from the macro, what we can see on a piece of paper, down to the micro, all the way to invisible traces of DNA. And that means the old way is super vulnerable, while the new way is highly resilient. We're essentially trading in a simple paper certificate for a biological fingerprint. It's a massive upgrade.
Okay, so that's it then, right? It seems like we've got our answer. DNA testing is the perfect solution. It solves everything. Case closed?
Well, not so fast, because as powerful as this technology is, and it is incredibly powerful, here's the important part. Just being able to detect something isn't enough to guarantee integrity across the entire system.
And this is where the real complexity kicks in. You've got this amazing lab evidence on one side and the goal of true food integrity on the other. But there's this gap in between, a critical bridge that has to be built. We can call it the translation challenge. All that amazing scientific data has to be translated into actual decisions by regulators, by auditors, and by the industry itself.
At the end of the day, it all comes down to building an architecture of trust. And that architecture needs two really strong pillars. The first one is that strong laboratory evidence, from science like PCR. But the other one is just as important, the strong systems, the regulations, the processes that actually use that evidence to protect all of us.
So what's the big takeaway from all this? Real, true food integrity isn't a choice between science and paperwork, it's about a seamless fusion of the two. To really get it right, we have to master both the molecule and the system that governs it.